Picture this. A revolutionary new drug, years in the making, poised to save countless lives. But there's a problem. A profound, almost invisible bottleneck that silently chokes off progress. Finding the right patients for the clinical trials.
It sounds simple, doesn't it? Just match the patient to the trial.
Exactly, but the reality is far more complex and, well, as we'll discover today, it actually costs lives and delays breakthroughs. Okay, let's unpack this. Today we're embarking on a deep dive into the intricate world of patient eligibility for clinical trials. Our guide is a fascinating 1993 research paper from Stanford University titled A Methodology for Determining Patients Eligibility for Clinical Trials. Now, this paper laid out a groundbreaking approach to an age-old problem.
And what's truly fascinating here is that even with the technology of the early 90s, these researchers zeroed in on a fundamental challenge that persists today.
Which is?
Which is, how do you efficiently and accurately match patients to the very specific, often numerous, criteria of a clinical trial? I mean, it's a task that's incredibly knowledge and data intensive.
Right. Demanding a level of precision that's almost impossible to achieve manually. It leads to this kind of inevitable human blind spot.
Absolutely.
So over the next few minutes, we're going to walk through the frustrating reality of patient recruitment, the dizzying maze a clinician has to navigate by hand.
Yeah, the manual struggle.
Then we'll uncover two innovative computer-based approaches proposed by this paper. We'll explore their ingenious synthesis, and finally, we'll reveal the surprising, almost shocking, real-world results that expose just how many opportunities were being missed.
Yeah, those results are quite something.
Okay, so at its heart, clinical trials are the absolute bedrock of modern medicine, right? They're how we rigorously evaluate new therapies.
To make sure they're not just effective, but also safe. Yeah.
And every single trial operates under a strict protocol document. Think of it as the trial's constitution.
It really is. It specifies everything from the study design, the interventions, to the crucial entry criteria, potential toxicities, how monitoring happens.
It's non-negotiable.
And if you don't find enough patients who fit those exact, often incredibly detailed criteria, well, the consequences are severe.
The paper gives an example, doesn't it?
It does, a stark one. It mentions none of the 10 clinical trials monitored by one division of the Veterans Administration could be completed on time, and 2 had to be terminated prematurely.
Wow.
And that was directly due to insufficient recruitment. Just imagine the scientific progress stalled, the potentially life-saving treatments that never reached patients.
All because of this one critical hurdle. That's truly devastating when you think about it.
It is.
It's not just about finding some patients. Many clinical trials require truly large sample sizes to achieve sufficient power.
Right. Statistical power, you need that power to detect statistically significant differences between, say, a new treatment and a placebo or standard care.
So without enough participants, you simply can't prove your case, even if a new therapy is genuinely revolutionary.
Which means all that research, all that hope can just fizzle out. Lost.
Now let's really step into the clinic back in '93, or frankly, even in many places today. If this is all done manually, how does a clinician even begin to untangle that web?
It's a mountain of information. Patient records, trial criteria. How do you match them without missing something vital?
The paper describes it as examining medical records and matching patient characteristics to specified eligibility criteria.
But that doesn't capture the sheer effort involved. It isn't a quick scan. It's a deep dive into handwritten notes, lab printouts, maybe different electronic systems.
A monumental detective job.
It absolutely is. And those criteria are far from simple. Just looking at Figure 1 in the source paper, it paints a vivid picture.
Can you get some examples?
Okay, so you might need a patient to have a specific diagnosis, like HIV positive or diagnosed with AIDS. Then you have laboratory results with incredibly precise ranges. Platelet count greater than 75,000 per cubic millimeter, or a CD4 count less than 300.
Very specific numbers.
Extremely. Even things like liver enzyme levels, SGOT less than 2 times upper limit of normal. Then you've got demographic criteria like age greater than 18 years.
And medication restrictions.
Oh yes, often very specific ones. Things like no therapy with rifampin, phenytoin, and a list of others within last 30 days. Or even no prior therapy with AZT for longer than 90 days.
Wow. Imagine trying to track all of that.
Yeah.
for multiple patients.
For multiple trials while also seeing other patients dealing with emergencies. I mean, it's a recipe for burnout and crucially for missed opportunities.
Right. And the clinician's actual thought process, which figure 2 in the paper outlines, makes it even clearer. It's not just checking boxes.
No, not at all. They start with basic requirements, then look at routine test results, but then it gets really dynamic.
Like what?
Well, they might realize they need to collect additional information that isn't readily available, or they might need to consider therapy adjustments to actually make a patient eligible.
So changing a medication, maybe?
Exactly. Or perhaps ordering unusual tests like a lumbar puncture, which isn't routine. And then there are subjective evaluations, things like assessing patient compliance, which is critical for a trial.
It's not a linear checklist. It's a multi-step, iterative process full of loops and forks.
That's precisely why the researchers underscored the challenges. The sheer number and complexity of the eligibility criteria mean that potentially eligible patients may be overlooked.
It seems almost inevitable.
Well, if we connect this to the bigger picture, you can see how this manual, intricate dance, especially when, as the paper says, dozens of clinical trials can be active simultaneously in a large clinic.
Yeah.
It almost guarantees that patients will slip through the cracks. It's simply too much information for one person to perfectly track all the time, no matter how dedicated they are.
Okay, so faced with this overwhelming manual maze, the Stanford researchers asked the critical question, could a computer actually help?
Could technology lend a hand?
This is where their core proposition really shines. The big idea was that a computer program could provide intelligent assistance.
How so?
By identifying areas of incomplete information, by indicating the possibility of a patient's condition changing, and by identifying characteristics that can be modified or acted on to render a patient eligible.
Ah, so it's not about replacing the clinician.
Not at all. It's about empowering them, helping them see the full potential within their patient pool.
Exactly. Providing smart, targeted support.
Yeah.
By automating part of this complex eligibility determination, a well-designed system can significantly reduce the information load on the clinician.
Which frees them up.
Right. Frees them up to be more systematic in identifying potentially eligible patients. Less time buried in paper, more time on patient care and recruitment conversations.
Makes sense. But to even build a system like this, the researchers first had to figure out how to translate those complex rules into something a computer could understand.
They needed a kind of digital language for eligibility criteria.
And what they came up with was, well, I think it's a brilliant classification system. They grouped criteria not just by what they were, but by key characteristics.
Like how objective they were, how much they varied over time, and crucially, whether a clinician could actually control them.
Which allowed the system to understand the nature of each requirement, not just its content. Very clever.
This classification is incredibly insightful because it moves beyond a simple yes or no. It helps the system understand the type of no it might be dealing with. They identified 5 main groups.
Okay, walk us through them.
First, stable requisite criteria. These are immutable preconditions, think permanent facts, like a patient's history of a specific disease or a known drug intolerance. They won't change.
Got it, the stable.
Second, variable routine criteria. These are things that change over short periods and are usually collected routinely. Lab counts, for example, CD4 count, platelet count, dynamic stuff.
Okay, variable. Makes sense.
Third, controllable criteria. These are circumstances a physician can actually modify. Like, if a protocol says no specific drug within 30 days, the physician could switch the patient to something else and wait, making them eligible. Active intervention.
Ah, so things you can potentially change.
Precisely. Fourth are subjective criteria, which rely heavily on a physician's expert judgment. This could be their estimate of patient compliance, likely survival duration, or something like the Karnofsky score, which gauges functional capacity.
More judgment-based.
Exactly. And finally, special criteria. These involve unusual, often costly or invasive tests, like that lumbar puncture example, things you wouldn't do unless the patient was already a strong candidate.
So this classification, it's more than just neat categories, isn't it?
Oh, absolutely. Here's where it gets really interesting. This isn't just categorizing. It's empowering clinicians to see the potential in a patient who might not be eligible today, but could be tomorrow with a small change.
How does it do that?
Well, 2 main ways. Prospectively looking forward, the program can see if a patient fails only on variable or controllable criteria. It can then alert the clinician saying, hey, this patient could become eligible, or Here's something you could potentially act on.
Like giving them a heads up on potential future candidates.
Exactly. And retrospectively looking back, this classification lets you analyze why patients failed criteria across the board. Are lots of people failing because of one controllable factor or because certain data is always missing?
That gives you powerful insights for improving the whole process.
Right. It pinpoints systemic issues or common roadblocks. This insight, even back in '93, was about using smarter information to help human decision-making.
Okay, so they classified the criteria. Now, the system needed a way to actually compute an eligibility score for each patient, right?
Yes, how to boil it all down. And the paper explored two distinct approaches for this.
Let's start with the first one, the qualitative approach.
Okay. This method used what they called discrete, non-numeric eligibility scores. Basically simple labels.
Like P for patient meets, PP for probably meets, N for datum not available, FP probably fails, and F fails.
Exactly, simple labels. And a critical part was how it handled missing data using these missing value assumptions.
Can you give an example?
Sure. Like, if a patient's record didn't specifically mention they were enrolled in another clinical trial, the system would just assume they weren't enrolled. Score P.
Makes sense, usually.
Or for something like a platelet count, if the value was missing, but the previous value satisfied the criterion within a reasonable time frame, it might assign PP for probably meets.
Okay, so heuristic rules, basically.
Right. And then these individual scores were combined using simple logic rules, like and/or truth tables shown in the paper. If a patient needed A and D, B and D, C to be eligible, and they got an F score for C.
Then the overall score was F failed.
Simple, logical. But they noted it involves sensitivity versus specificity trade-offs. You could tweak the rules to catch more potential candidates.
But you probably also flag more people who weren't actually eligible, creating more work for the clinician to review.
Precisely. That's the trade-off.
Okay, so that was qualitative. What was the second approach?
The second was the probabilistic approach. This took a much more, let's say, mathematically rigorous route. It introduced Bayesian belief networks.
Okay, Bayesian networks. Can you explain those briefly?
Sure. Imagine a map, like a directed graph, where nodes represent different variables, patient characteristics, lab results, criteria, and the links show dependencies between them.
How one thing influences another.
Exactly. This approach calculates an eligibility score as a numeric probability, a number between 0 and 1. And the clever part is that observed patient data, the things you do know, can dynamically update the probabilities for the unobserved criteria, the missing data.
So it gives a more nuanced picture than just probably meets.
Much more. And the key difference. This approach can compute the actual probability that a patient satisfies all the eligibility criteria simultaneously, taking those dependencies into account.
Ah, okay. So comparing the two, they both represent criteria, define scores, handle missing data in some way.
Yes, similarities there, but crucial differences too. Missing data handling, for example. The qualitative use those heuristic rules, which can be hard to verify for consistency.
Whereas the probabilistic approach,
It used formal probability distributions, consistent by design, and the summary scores were fundamentally different.
how so?
That PP probably meets in the qualitative summary does not have a probabilistic interpretation, as the paper states. It often just reflects the lowest score in a conjunction. The probabilistic approach gives you a true probability of meeting all criteria together.
That sounds much more rigorous. But what's the catch?
Cost and complexity. Building and running these Bayesian networks is complex. It's computationally intensive, what computer scientists call NP-hard.
Meaning it gets very slow as things get bigger.
Right, and you have to meticulously model all the dependencies, like how Karnofsky's score might relate to liver function, and get experts to estimate those probabilities. The qualitative approach was much simpler, much faster computationally.
So what does this all mean for the clinician on the ground? It's a trade-off, isn't it?
Exactly. It's a trade-off between a simpler, quick assessment using the qualitative rules and a more rigorous but computationally complex and harder to build probabilistic one.
Given these sort of complementary strengths and weaknesses, the paper then proposed, well, merging them.
Yes, a combined approach. They saw the value in trying to get the best of both worlds.
How did that work?
Okay, look at figure 6 in the paper. The idea was to use the Bayesian network structure for modeling the individual criteria and handling the missing value assumptions. This gives you that uniform and consistent representation and a principled way of computing beliefs or probabilities for each single criterion.
So use the rigor of probability for the individual parts.
Exactly. But then, before combining them into an overall summary, the system maps those calculated probabilities back to the simple qualitative scores PPPN, FPF.
Ah, I see. Back to the simple labels.
Right. And then it uses the straightforward and/or logic tables from the qualitative method to calculate the final summary eligibility status.
Clever. So you get the probabilistic rigor underneath, but the simple summary on top.
Precisely. And the benefit, as the paper points out, is that this avoids the need to make any assumptions of dependency among the eligibility criteria when calculating the summary score, because you're combining the qualitative labels, not the raw probabilities directly.
Which keeps the final output intuitive for clinicians. It still has those clear semantics defined by the simple logic tables.
Exactly. Best of both worlds in theory.
Okay, now let's bring this discussion from theory to the tangible. Did they actually build this?
They did. They implemented the qualitative approach in a system called Therapy Helper at Santa Clara Valley Medical Center. This was back in 1990, used for 17 HIV and AIDS protocols.
And they tested it?
Yes, they conducted a retrospective study. They looked back at the records of 60 HIV-positive patients covering 301 visits over a 7-month period.
And what did they find? Did the system work?
Well, the findings were truly eye-opening. The study concluded, and I'm quoting here, that clinicians inadvertently missed a significant number of opportunities for enrolling patients in clinical trials.
Significant.
20%. 1 in 5. That's 12 out of those 60 patients who were found to be provisionally eligible for at least one protocol during that period, but were not enrolled.
Wow, 20%. Think about that impact.
It's huge. Patients who could have potentially benefited, potentially contributed to vital research, just overlooked. The insight here isn't just that tech can help, but that the sheer cognitive load of the manual system guarantees missed opportunities.
It's a systemic challenge, not about blaming individual clinicians.
Absolutely not. It's about the complexity overwhelming human capacity. This paper revealed that crucial human element back in '93, proving that even with the best intentions, information overload hinders science and patient access.
That's astonishing. And it wasn't just about overlooking patients who were clearly eligible, right?
No. The study also found that 17%, 10 of the 60 patients, satisfied all criteria except for controllable ones.
Meaning a physician could potentially have intervened.
Yes. Maybe adjusted a medication, waited a specific period. Simple actions could have made them eligible. Opportunities for action were missed.
And was missing data a problem?
A big one. The data needed for eligibility checks was frequently missing. Platelet counts were absent for 31% of visits. CD4 counts for a whopping 43% of visits.
So the system couldn't even make a determination sometimes.
Exactly,
Huh.
which just hammers home the value of an automated system. The paper concluded that an automated eligibility screening program could increase patient accrual by alerting clinic staff to those data that should be collected and to those criteria that can be manipulated.
It flags the gaps and the potential actions. This raises an important question. How many more patients could have benefited from these trials, and how much faster could research have progressed if this system had been widely in use?
It's a sobering thought.
And the implications of this methodology, this way of thinking about eligibility, extend even beyond clinical trials, don't they?
They absolutely do. The paper draws a really powerful parallel to medical practice guidelines.
Okay, explain that connection.
Well, practice guidelines are essentially standardized specifications for patient care based on the best available evidence. They tell clinicians, for a patient like X, the recommended approach is Y.
Right, standardizing care.
But clinicians trying to apply these guidelines face very similar challenges to trial recruitment. Is the guideline actually applicable to this specific patient? Often crucial data might be missing from the record.
Or the patient might have a condition that technically rules them out, but it's something controllable.
Exactly. The same issues of missing data, variable conditions, and controllable factors apply. So this methodology developed for trials.
Could be applied to the problem of determining whether practice guidelines are applicable in a given clinical situation.
Precisely. It could help ensure guidelines are applied more consistently and appropriately, potentially revolutionizing how routine patient care is standardized and improved based on evidence.
What an incredible exploration we've had today. We've seen how a computer-assisted eligibility system, conceptualized way back in 1993, could have dramatically improved trial recruitment.
By identifying those missed opportunities, flagging missing data, and highlighting those controllable factors.
And beyond trials, this whole methodology holds immense promise for enhancing how we apply medical practice guidelines in everyday care.
It's truly transformative potential, and it makes you think. Consider the journey from that 1993 paper with its, early 90s computing power and relatively simple rules.
To today's sophisticated AI capabilities.
Right. If these early systems could identify a staggering 20% missed opportunities for trial enrollment, just imagine. What could truly integrated, intelligent systems powered by modern AI do for patient care and medical research now?
That's a really provocative thought to leave our listeners with. It really makes you reflect on the powerful synergy possible between human expertise and technological assistance.
It's not about replacing the clinician's judgment or empathy.
No, it's about augmenting their incredible knowledge and dedication with tools that can sift through immense complexity, highlight the unseen, and ultimately accelerate medical breakthroughs for all of us.